I got really lucky with when it was like, I got a friend. I got so lucky. He was not home last money when I was
Okay,
when I got to that.
so let's quickly do this. So we are looking at muscle tissue, and it's sort of the first thing I wrote. Everything got wiped out, so I'm going to rewrite everything again. So I had mentioned how the three muscle tissue we have is cardiac, skeletal, and smooth. And we had mentioned how cardiac muscle is only found where? In the heart. Skeletal muscles are all muscles that we can consciously what? Control. Okay, we can consciously control. And then smooth muscles are muscles that we cannot consciously control. Okay. So, and also I mentioned how skeletal muscle is usually attached to what? Bones. Okay, it's usually attached to bones. All right, but that doesn't mean all of them are, you find them attached to bones. For instance, the sphincters, some of the sphincters, like the external anal sphincter, is made up of skeletal muscle, but it's not attached to a bone. So smooth muscle, we cannot consciously control, just like cardiac muscle, but smooth muscle are not cardiac muscle. They are muscles like the stomach muscle and intestinal wall muscle and blood vessels, the muscles on the walls of the blood vessels. So today what we are going to concentrate on is just skeletal muscle what? Tissue. All right, that's what we are going to concentrate on, the skeletal muscle tissue. And we are going to look at the anatomy and what? The physiology. Okay, the physiology component, you are going to concentrate more on how the muscle contracts. Okay, so the anatomy part is how does it look like? How does the muscle tissue look like, and how was it formed? So as I had mentioned earlier, there are these cells called myoblast cells, and these myoblast cells tends to what? Fuse together. Okay, so let's say three of these myoblast cells, that's what I had mentioned, had fused together to form the muscle fiber. Okay, so they form what we call the muscle fiber. So since it's three cells, we'll have how many nuclei? Three nuclei. That's why the muscle fiber is also multinucleated. And then one of the cells will form the microsatellite cells or myosatellite cells. And these satellite cell is important to repair the muscle fiber when there is any damage. Does that make sense? Okay, so every cell contains cytoskeleton, and we mentioned how the cytoskeleton is made up of all these filaments. And so this is what happens. As a muscle fiber is maturing, it will be, the inside of the cell will be filled with filaments, the cytoskeleton filaments. It's going to be filled with filaments, and these filaments are made up of different types of proteins we'll be looking at. So the filaments are arranged within the cell. The filaments are arranged into what we call myofibrils. Okay, myofibrils. So let's look at how that looks like. So let's look at how that looks like. This is a myofibril. Okay. Another what? Myo what? Fibril. Another what? Myofibril. Are you all getting this? And these myofibrils are filled with what? Myo what? Filaments. Are you all getting this?
Yep.
Okay. So these myofibrils are located where? Inside of what? The muscle what? Cell. Okay. So the muscle cell, the other name for, you realize that, for instance, the other name for plasma membrane is called plasma lemma. All right, just so have this at the back of your mind. I'll mention something to you shortly. Lemma. Okay. The other name for plasma membrane is plasma lemma. So just have this at the back of your mind. I'll mention that later on. So anyway, let's look at how we are going to look at how one myofibril will look like. So if this is A myofibril. Inside of that one myofibril, we are having what? Filaments. Are you with me? So these filaments are arranged nicely. They are arranged into units of what they call sarcomere. Okay? Yeah, the filaments inside. So it means a myofibril is made up of what? A lot of what? Sarcomeres. And we are going to look at how a sarcomere, one sarcomere will look like. All right? So anyway, just to give the overview, let's continue. Now, it means that in a myofibril, we are having what? Sarcomere connected to another sarcomere, connected to what? Another sarcomere, on and on. Are you with me? Now, surrounding, so these myofibrils are all inside where? The muscle what? Fiber. So the plasma membrane of the plasma membrane of the muscle fiber, instead of calling it plasma membrane, guess what they call it? They call it sarcolemma. They call it sarcolemma. Are you with me? Now you need to pay attention to this. So the plasma membrane is called what? Sarcolemma. And this plasma membrane has a connective tissue wrapped around it. This connective tissue wrapped around the muscle fiber or the muscle cell. The other name, either you call it muscle fiber or muscle cell. Same thing, all right? So each muscle fiber or muscle cell has a connective tissue wrapped around it. That connective tissue is called endomysium. Are you all getting this? Okay. So let me have this over here. This is what? Endomysium. Watch this closely. Another word. Endomysium. Another word. Endomysium. Are you all picturing this? Okay, inside this endomysium, what do you have? The myo what? Fibril. You have what? Myofibrils. Myofibrils. Okay, and since the myofibrils are inside the muscle fiber, they will have what? A cell membrane around them, which we call what? Plasma lemma. Does that make sense? So this is a muscle fiber, and inside the muscle fiber you have what? The myofibrils. Does that make sense? Are you all getting this? And then the myofibrils have what? The myofibrils are located in the muscle fiber. They have the plasma membrane, which they call what? Plasma what? Lemma. And then the plasma lemma is surrounded with a connective tissue called what? Endomysium. Capiche? Okay. So each of these will be what? One what? Muscle fiber or muscle cell. So this is a muscle fiber. Are you all getting this? Now another connective tissue can wrap around a group of what? Muscle fibers. This connective tissue is called perimysium. Okay? So this connective tissue is called what? Perimysium. So this a group of muscle fibers that have a connective tissue wrapped around them, this whole thing will now be called a fascicle. Okay, a fascicle. So we can have fascicles. Okay, another what? Fascicle. Do you all see that? Okay, so these are all what? Connective tissue wrappings called what? Perimysium. Are you all getting this? So again, located in the perimysium, we are going to have what? The muscle cell, right? Muscle cells. And these muscle cells contain what? Myofibril, 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 myofibril. Are you all getting this? Myofibril, myofibrils. And these myofibrils contain what? The filaments, or known as myofilaments. And each muscle fiber has what? Endomysium what? Wrapped around it, endomysium wrapped around it, endomysium wrapped around it, endomysium wrapped around it, endomysium wrapped around it. Are you all getting this? Okay, endomysium wrapped around it. And now there is another wrappings that can wrap the whole fascicle. Do y'all see that? There's another connective tissue that can what? Wrap a group of what? Fascicles. And this connective tissue wrapping the group of fascicles is called epi what? Mysium. Okay, so that gives us the whole what? Muscle what? Structure.
So go from the sarcolemma to plasmalemma with the endomysium to the fascicle.
So the filaments are arranged into what we call what? Sarcomere. And a lot of filaments come together to form what? A lot of the filaments or sarcomere units will come together to form what? Myo what? Fibrils. And a lot of myofibrils will be located where? Inside of the cell, muscle cell, or the muscle what? Fiber. All right, and the muscle fiber will have what wrapped around it, and the mysium what? Wrapped around it. And a group of muscle fibers will come together, and they will have what? Perimysium wrapped around it and form what we call the fascicle. And then a group of fascicles will get what? Epimysium wrapped around it to form the whole muscle tissue.
So it just keeps
It keeps layers, layers and layers and layers around it. Okay. So now, so this is just a general overview of it. So as I had mentioned to you, the muscle tissue is made up of cardiac, smooth, and what? Skeletal. And this is showing us how the skeletal muscle, how it looks like. The cardiac muscle, it's actually branched and it's connected with what we call the intercalator disc. And this is the smooth muscle. So basically, let's look at the general function of the skeletal muscle. The skeletal muscle is important because most of them is attached to the bones. What connects skeletal muscle to the bones? Tendons. Remember the connective tissue called tendons. There are certain tendons that are flat, and they call them aponeurosis. Okay, aponeurosis. We'll look at them later on. So muscle also helps to give us our posture. So that's why you see all these weightlifters. They have their hands hanging next to them. Okay, so it gives us our different postures. For instance, our six-pack muscle, the rectus abdominis muscle is covering and shielding visceral organs like the organs in our abdomen, so supports soft tissues. And like I had mentioned, sphincters. Are like the anal sphincter controls when we want our poop to come out or not. But of course, if you have diarrhea, we have two types of sphincters: the internal and external. The external is made up of skeletal muscle, so you can consciously control it and close your anus. But the internal sphincter is made up of smooth muscle, so it will get its instruction from your body directly and not from your brain what you want it to do. So with the internal sphincter, it will actually relax when it senses poop in there. That's why when you have diarrhea, it will relax on you. And then you are trying to close with the external and it's not closing, so you better run to the back. So although they are both sphincters, one is skeletal muscle, one is smooth. So they guard entrances, okay, and exits, some parts of them. And when we are cold, now that the weather is cold, when you are very cold, you find yourself shivering, right? So it helps with body temperature and stuff like that. And to interest you, those of you are marathon runners, glucose, excess glucose in our body can change and form what we call glycogen. Okay, glycogen is not only stored in the liver, it also gets stored in the muscle as well. And so it provides nutrients as a nutrient reserve for energy in case we need it. And so when you look at a whole skeletal muscle, just picture meat, right? Just picture meat, chicken or pork or something like that. You see the muscle fiber, which is the meat. There's connective tissue, which are all these wrappings and fascia, and some of the epimysium and some blood vessels and nerves are all over on there. So what are some of the connective tissues that we see? Of course, the tendon, which connects to the bone. Like I mentioned, for instance, the skull. We have the muscle over here called frontalis occipitalis. You do that when you're in the lab, you talk about it. So the tendon that holds these muscles on the skull is flat. It's not rounded like it's in the form of a sheet. So they call that aponeurosis. Does that make sense? Okay, so this is what I was telling you about the wrappings. And I like to use this I like to use this PowerPoint slide to show you what I just explained. This other PowerPoint slide to explain to you how the muscle looks like, which I've already explained. So as I mentioned, we have what? The filament, right? And a lot of filaments arrange themselves into what we call the sarcomere to form what? The myofibril. Do you all see that? So a muscle fiber or a muscle cell, this is what? One what? Muscle cell. Do you all see that? It's filled with what? What? Fibrils. Does that make sense? Are you all picturing that? So what is it? And the membrane of the muscle cell is called what? Sarco what? Lemma. This is what they call the cytoplasm. Sarco what? Plasm. Do you all see that? And the filaments are arranged very nicely and they form these striations. That's why we say that the skeletal muscle is what? Striated. And we'll look at the arrangement, which is what we call the sarcomere arrangement. And wrapping this whole muscle cell or muscle fiber is the connective tissue wrapper we call what? They call what? Endomysium. So this is what? Endomysium wrapping around each muscle cell, muscle fiber. Do you all see that? And they also get another sheet wrapping what? Around it, and that sheet is called what? Mysium. And a perimysium with the muscle cells in there is called what? Fascicle. Okay, it's called what? Fascicle. So this is the fascicle. So we have a fascicle. Okay, another what? Fascicle, and all these fascicles come together, and another what? Wrapping, and the connective tissue wraps around it, and they call that what? Epimysium. Okay, they call that what? Epimysium. Does that make sense? Okay, so that's what I was saying. That's what forms the muscle. Okay. Okay. So this is what I was telling you about how muscles are formed. We have these, we call them what? Myoblasts, and they fuse together to form the skeletal muscle what? Fibers. So each skeletal muscle fiber, the nucleus came from where? The myoblast. So that's why they are multinucleated. So this is showing us the myoblasts fusing. It's not all of them that fuse, so they form what? Myosatellite cells, like I mentioned. And these myosatellite cells are important for repair. So these myosatellite cells, this is showing us an immature muscle fiber. It will mature because a lot of filaments will be in there, and the filaments will be what? Nicely what? Arranged, and that is what will give the striations we see in there. Okay, so like I said, the plasma membrane is now called what? Sarcolemma, and it can contain a lot of what? Nuclei, because they are being fused and they can be very long as 12 inches. And the cytoplasm is now called what? Sarcoplasm. So because they are multinucleated, they have a lot of genes, they can perform a lot of function in a faster way. That's why muscle just contracts just like that. And for them to contract, it means they need what? A lot of energy, and the powerhouse of the cell is what? The mitochondria, okay, which will help produce a lot of it. So we'll keep looking closely at it. So we are going to look at what? The myofibrils. Like I said, the myofibrils are made up of what? Protein what? Filaments, right? And these filaments, we have thin and what? Thick filaments. Majority of the thin filament is actin, but it also has what we call tropomyosin and troponin, which we'll talk about troponin. Okay, and the thick filament is mainly myosin, right? It also has other proteins as well, which we are going to talk about shortly. So let's look at it again. This is a myofibril. So one muscle cell is filled with what? This is a sarcolemma, sarcoplasm, and it has a lot of what? Myofibrils. So these myofibrils, let's look at it closely. It's made up of what? Thin and what? Thick filaments arranged nicely. Do you see how they are nicely arranged? Okay, and this represents a sarcomere. So they are arranged into sarcomeres. But watch this closely. Let's look at this. So this is a section of what? The muscle what? Fiber. Do you see there are holes on the sarcolemma, which is the cell membrane? Do you all see that? Watch this closely. Let's look at this hole over here. Do you see this hole? It's a continuation. Do you see this hole? It continues into these tubes. Do you all see that? So these tubes you are seeing wrapped around the muscle fiber they are all what? Continuation of what? The cell membrane. In other words, when I pour a green paint through this hole, I pour a paint through this hole, it will color this whole space. Does that make sense? Do you all get this? Okay, we'll see the importance of these plasma membrane extensions wrapped around the myofibrils. And these tubes wrapped around them are called transverse tubules or T-tubules. Same thing. Okay, and it's important because when an electrical impulse touches a receptor on the cell membrane, it can allow the transmission of the impulse through the myofibrils so that they can contract. And then it will allow the whole muscle to contract. Without these holes in there, the only filaments that will slide against each other for contraction to occur will be the filaments that are what? Close to the surface of the what? Plasma membrane. Does that make sense? So in order to reach the ones in the middle as well, you need the plasma membrane to be close to them, and the way to bring the plasma membrane close to them is to create these T-tubules to wrap around these myofibrils. Does that make sense? Because they are continuous with the plasma membrane. Do you all get this? Okay, good. So apart from these T-tubules, do you see there are these blue sac-like structures wrapped around each myofibrils? Do you all see that? This is the endoplasmic reticulum. It's what? The endoplasmic what? Reticulum. Remember, it's an organelle in the cell, right? Now, we have two types of endoplasmic reticulum. Do you think which one do you think this one is? No. Is it rough or smooth? Smooth. Why? Because it has no ribosomes attached. Good. So it's smooth endoplasmic reticulum. And guess what? It's been modified. This SER, smooth endoplasmic reticulum, has been modified to store calcium. Do you remember when we were doing the bones, I told you that calcium is needed for the muscle to contract? There it is. We'll see how it plays its role shortly. So this smooth endoplasmic reticulum, the aim is for it to store calcium, because we see how calcium is important for muscle contraction. And guess what? Instead of calling it smooth ER, they now call it sarcoplasmic reticulum. Okay, they call it sarcoplasmic what? Reticulum. All right. Okay. And of course, there's a whole lot of what? Mitochondria all over in order to give what? The energy that is needed. Okay, good. So let's zoom in and picture how the filament, how it looks like inside a sarcomere. Okay, so like I said, um, the myofibril is made up of what? Units of what? Sarcomere. And these repeating units of sarcomeres is what allows the muscle to what? Contract. So for instance, certain cells have roughly certain cells, one myofibril can be made up of roughly how many sarcomeres? 10,000s of what? Sarcomeres to form one myofibril. So let's look at how the I'm going to, you see all these Z lines having actinins, A band, I band, M line, H band, all these things. I'm going to explain it to you right here. All right. So pay attention to this. We are going to look at one what? Sarcomere boundary. Are you all picturing this? Okay. This part, the proteins that form these Z-looking things, these proteins are called actinides. Okay, these proteins are called what? Actinides. Are you all with me? Okay. Are you paying attention? These proteins are called what? Actinides, and this, the boundary of a sarcomere is called the Z what? Line. Okay. Now attached to the Z line, you see this coming off it? Do you all see this? Okay. This light reddish thing attached to the Z line. This is what? The thin what? Filaments. Okay, they are what? The thin filaments. Now watch this closely. While these purple ones are what? Thick filaments. Okay. So now that we've identified, so this is the boundary. Okay. Now, where the thick filament traverses, do you see here? Where the thick filament traverses or occupies is called what? The A band. Now, this is, be quiet, please, too much chit-chatting going on at the back. These thick filaments that occupies this, where the thick filament occupies is called the A band. This A band could have thin filaments as well. But again, it doesn't matter. What is important is the area where the thick filament occupies is called what? The A band. So the length of the A band cannot change. Does that make sense? Because it's the area where what? The thick filament occupies, irrespective of whether there's a thin or filament or not. Does that make sense? Okay, now there is the middle part of the thick filament is held in place. We'll mention the proteins that help that. And that middle part is referred to as the M line. Are you with me? So what helps to suspend the thick filament is these proteins in the middle called what? The M line. Now, what is the I band and what is the H band? The I band is the area where there is only thin filaments. So the area of the sarcomere where there's only what? Thin filament is called what? I band. And then the H band or the I zone, the H zone or we call it H zone or H band, is the area where there is only what? Thick filament. Okay, the area where there's only thick filament is called what? H band or H zone. And the area where there's only what? Thin filament is called what? I band, or sometimes they call it what? I zone. So that's a part of what? The sarcomere. That's a part of what? The sarcomere. So I'm going to show you a closer look. So this is what? A myofibril. These are the filaments in there. This is what? The sarcoplasmic what? Reticulum. This is the T tubules. All right. This is the sarcolemma. And like I said, they call it. So I'm going to mention something to you about the sarcoplasmic reticulum. This is what I was telling you. This is the cytoplasm of the muscle fiber called what? Sarcoplasm. This is the endoplasmic reticulum. They call what? Sarcoplasmic reticulum. So energy, these are pumps, calcium ion pumps. Energy is used to pump calcium from a low concentration in the sarcoplasm to what? High concentration in what? The sarcoplasmic reticulum. And we have, so the pump keeps doing that and it uses energy to do that. But we have channels, calcium channels that are gated, so they can be closed or open. So anytime calcium is needed for the muscle to contract, the gate will open and the calcium will what? Come out. All right. And we are going to look at how that happens. So let's go back here. Okay. So again, just in summary, the sarcomere is made up of what? Boundaries called the Z line, which contains what? The actinin proteins, and then we have what? The thin filaments, right? Attached to it. And then we have what? Thick filaments suspended where? In the middle by what? The M line. So this is what happens. You see these thick filaments have what we call, most of it is made up of myosin. They have what we call myosin heads. Okay, and this is how the muscle contracts. Okay, these myosin heads will attach to the actin. So watch this closely. I'm going to use a myosin head to attach. Do you see it as attached to the actin? Are you all seeing this? And then it's going to pull the actin towards each other. Do you all see that? So when it pulls it so we call this the sliding filament theory. That's how our muscle contracts. It will pull the actin what? Towards each other, and what will happen to the area of the H zone is going to get narrower. Does that make sense? And the area of the I band is also going to get narrower because this will keep overlapping with what? The muscle. Sorry, the myosin, the thick filament. Does that make sense? Okay, so let's look at this picture over here to throw more light into this. Okay, this is what? The I band, right? And this is what? The this is H band, this is what? The I band. Are you all seeing this? So the H band is narrower. Now you see all these little myosin heads, they hold the what? Actin and pull it what? Towards each other. So as it pulls it, what do you think will happen to the overlap? There will be more overlap between what? The thin filament and what? The thick filament, because it's moving in this direction. Does that make sense? So you can see that there's more overlap between the thin and what? Thick filament. Does that make sense? So what is going to happen to the area of the I band? It's going to get what? Narrower, and the area of the H zone as well. Does that make sense? So this is a sarcomere at rest, and this is what? A sarcomere which has contracted. So that's how our muscle contracts, and we are going to look at how it actually does that specifically. How does that happen? We'll look at that specifically. So let's go back to where we were. Okay. So again, sarcolemma is what? The plasma membrane. Inside is what? Sarcoplasm. It helps to transport transmembrane potential, which brings about impulse. So when I tell my bicep muscle to contract right now, what just happened? Contract. I have contracted it. What just happened? Neurotransmitters travel through my brain through nerves attached to what? Receptors on the plasma membrane, plasma lemma of my muscles, and send what? Action potential or impulse through the T tubules to the microfilaments for them to slide. How did they do that? What did the neurotransmitter specifically do to allow the filaments to slide and contract. These neurotransmitters cause what we call an action potential or an impulse to be what generated, and that impulse causes calcium to what come out of the sarcoplasmic reticulum, and calcium helped the process of what contraction. What did calcium actually do? That's what I'm about to tell you. How did calcium help the muscle five filaments to slide against each other? That's what I'm about to tell you. Do you all get the picture now? Okay, so of course, for these filaments to slide, so myosin head will hold the thick filament and then they'll slide like this. For that to happen, energy is needed. So we are going to see how that happens as well. Does that make sense? We are also going to look at exactly where these myosin heads, which part of the actin they are attaching to, and how they are bringing the contraction. So we are looking at it in that detail. Isn't that cool? Isn't it? The human body amazing? Right? All this is happening inside of you. Okay. So I had already mentioned to you the transverse tubules, right? They are what? Extensions of what? The sarcolemma, traveling inside the interior to encircle the sarcomeres. And then the sarcoplasmic reticulum, I've already mentioned to you, is what? The modified smooth endoplasmic reticulum. Okay? And it contains a lot of calcium. So like I mentioned, we have the pumps pumping what? Calcium inside from the sarcoplasm. So guess what? Energy is needed because the inside of the sarcoplasmic reticulum has 40,000 times more calcium in there than the sarcoplasm. So energy is needed for that to be happening, which is what I explained to you. All right. So in order for us to see how the sliding is occurring, we have to look at how the thin filament, how does it look like, and how the thick filament look like as well. So first, we'll start with the thin filament. Remember how we said the thin filament is attached to the Z line, and it's what holds it is what? The actin. So first, what is the actin? We have two types of actin, the G and the F actin. So the G actin is that site where the myosin is going to bind to in order to what? Move what? The thin filament. Okay, so remember I said if this is the thick filament and this is what? The myosin heads. And this is what? Z line, Z line. This is the thin filament. What happens? As I mentioned, the myosin head will bind to it and what? Drag it. Does that make sense? Drag it in this direction towards each other. Are you all getting that? So where exactly does the myosin head bind to on the thin filament? The part it binds to is what? The G actin. Okay. So, and then the thin filament has also what we call what? Tropomyosin and what? Troponin. This is how I explain this. Think of a pot, a shallow pot in your kitchen. This is a shallow pot in your kitchen, right? A saucepan in your kitchen. Okay. And or a frying pan, right? Yeah, frying pan may be better. So this is the G protein, the G actin, all right? And watch this closely. This is a lid. The lid here is tropomyosin. Are you all getting this analogy? Okay. This part is what? This part is the G what? Actin. And then this is what? The lid covering what? The G actin. And the lid covering the G actin is what? Tropomyosin. Are you all getting this? And this is troponin, which is pretty interesting. Yeah, the knob on the top. But there's three parts. So troponin, right? So the troponin somewhat has these. Okay, so do you see what I did? Okay, so the troponin is attached to what? G-actin. Do y'all see that? It's attached to what? Tropomyosin. Do y'all see that? And this part, the top part of the troponin will eventually bind to what? Calcium what? Ions. This is the best analogy I can give you. So, picture my fingers as calcium. Okay, so this is a pan, right? So picture my fingers as calcium. So what color should I use? I'll use this. Picture my fingers as calcium. So I use my fingers to what? Remove what? Hold what? Troponin. So my fingers will be calcium. Hold troponin, and what will I do? I'll displace what? Tropomyosin and expose what? The G-actin. So when I expose the G-actin, then the myosin head will bind to what? The G-actin on the what? Thin filament, and what? Pull it and close it. So that's how the contraction occurs. So we'll look at it again using this. Okay, as I mentioned to you, this is what? The Z line made up of actinin, and these are what? The thin filaments. You all see that? What is a thin filament made up of? It's made up of G-actins. So these are what? The G-actins. Do you all see that? It's made up of what? F-actins. Okay, the F-actins are in there as well. Okay, what holds the G-actin and the F-actin is what we call the nibulin protein. Okay, and now this is tropomyosin. This blue thing is what? Tropomyosin, and the tropomyosin is covering the, you see, we are having, if this is F-actin, sorry, if this is actin, we have the site, you see the yellow site is the G-actin area, while the other color is what? The F-actin area, which what holds them together, these two groups of strings together, which is F-actin, and this whole thing is made up of F-actin and what? G-actin, F-actin and G-actin, and what is holding them together is what? Nibulin. Okay, the nibulin. So let me clean this. So the tropomyosin is covering what? The G-actin. Do you all see that? Okay, this blue thing is what? Tropomyosin, and it's covering what? The G-actin. Are you all seeing that? And then the troponin, we have three areas. The part that is attached to what? The G-actin, the part that is attached to what? The tropomyosin, and the part that will eventually attach to what? Calcium. Does that make sense? Okay, do you all get this? The troponin is attached to three parts, okay, which is what? The tropomyosin, the G-actin, and what? Another part that is yet to attach to what? Calcium. So that's what I showed you with my analogy of the lid. So this is the troponin, which has what? Calcium, which is my hand. And you're going to see how calcium is what will lift the whole lid off. So this troponin is attached to what? The G-actin, right? It's attached to what? The G-actin. You all see that? Like what I showed, this is troponin, and part of it is attached to what? The G-actin. And then it's also attached to what? Tropomyosin, and this top part is going to be attached to what? Calcium. But as soon as calcium, which I said, picture it as your fingers, attaches to troponin, it's what? Raises the lid off and exposes what? the G actin. All right. Okay, that's the best analogy I can give you. So, before we move on to what the thick filament is going to look like, I want to show you how the contraction occurs using a different PowerPoint slide that I have. Okay, so this is what happens. This stands for what? Calcium. Do you all see that? The purple. So the calcium what? Attached to what? Troponin. Are you with me? And displace the tropomyosin. This is what? Tropomyosin. This is what? Troponin. Okay. This is tropomyosin. This is what? Troponin. And this is what? Calcium. Okay. Okay. So, and this is what? The myosin, and this is what? The myosin head. Do y'all see that? So, so what is going to happen is that ATP is going to come, and this ATP that comes, the myosin head will catalyze it and break it into what? ADP and what? Phosphate. And that will cause what? Energy to be what? Released. Do you remember how I told you energy is trapped in the ATP? So that energy that gets released will be used will energize the myosin head so that the myosin head can bind to what? The actin, like you can see it's doing over here. So when the ATP attached to the myosin head, watch, when the ATP is attached to the myosin head is not attached because ATP has the energy inside of it. It hasn't yet released the energy. The only time that this myosin head will have energy is when it breaks the ATP to open it up. Does that make sense? So when it breaks the ATP, when it splits the ATP to open it up and it forms ADP, it helps to orientate the head properly, and that energy is used to what? Attach to what? The actin, and the specific actin is what? G actin. And the specific area of the G actin is called the myosin binding. Someone is hitting my table. The myosin binding site. The specific part of the G actin which it binds is called what? Myosin what? Binding site. Are you all getting this? Okay. So when it binds to it, then now it will what? Pull it, drag it, like I showed you. Okay. And of course, as the myosin head rotates and pulls the filaments together in that way, what happens? The ADP will what? Of course, fall off because the ADP has to fall off to go and pick up more what? Energy and form ATP. Okay, so then it will use that energy to pull it. So when the myosin head has used the energy, when the ADP falls off, and now the myosin head has used the energy to what? Pull the thin filament. ATP needs to come to keep energizing what? The myosin head. Does that make sense? So ATP will come and bind to the myosin head. So as soon as it binds to it, the myosin head will what? Release, will detach. Does that make sense? So that it can catalyze the ATP and break it open again for the whole cycle to continue. So as soon as the ATP binds to the myosin head, that it detaches from what? The thin filament. Does that make sense? Now, if calcium is no longer present, if this calcium is no longer present, what do you think, if it gets removed, what do you think will happen? The tropomyosin will cover the G actin part. And now myosin head can have all the energy it has, but that place has been covered, so it cannot bind. So then the muscle will stay relaxed. Does that make sense? So that's how contraction of the muscle occurs. we will look at, I wanted to show you this on this PowerPoint slide that I have. So let's go back to our slide. Um, and now look at what the anatomy of it. So this is thin filament. How does the thick filament, how does it look like? A thick filament, of course, is made up of myosin, okay, myosin molecules, but it also have these proteins we call titin. And it's these titin proteins, if this is the Z line, and this is the myosin, okay, suspended in the middle with the help of the M protein. We also have these titin proteins, these springy-like proteins called titin proteins that helps to what? Connect the myosin head to the Z line as well. So they call them what? Titin proteins that help to, they are like elastic and they can recall and stretch. All right. And so the myosin head, the myosin is like wrapped, it's a wrap of things looking like this. Okay, so this is the head and these are the tail part. So it's a free head that usually binds to the actin. All right, so I've already told you about the sliding filament theory. This is showing us what I said, if it's relaxed and when it's what? Contracts. Okay, so this is showing us a myofibril, which is made up of what? Sarcomere, sarcomere, sarcomere. Do you all see that? And when it's relaxed, this is how it looks. When it contracts, it shortens. Okay, so the next time we meet, I'm going to start with the neuromuscular junction. This is focusing on how the neurotransmitter is what? Released and attaches to what? Receptors on our cell membrane, which will be what? The plasma membrane. Are you with me? And then how it brings about what we call impulse or action potential. Action potential, you have to do it in anatomy 2. You are supposed to actually do it in anatomy 1, and they moved it to 2 in this school. So it produces action potential, and it's this action potential that will cause the calcium to be what? Released. So we are going to look at muscular junction and continue the story from there. I think this is very interesting.